Hello, everyone. This is Ezrin Zaimi from the Faculty of Health and Life Sciences, Department of Diagnostics and Allied Health Sciences. So today's session with me is on the viruses, which is a very common topic nowadays and in our current times. So I hope you enjoy. Viruses cause most of the diseases that still plague the industrialized world, like the common cold, the influenza, herpes, AIDS, and to name a few. Although we have immunization against many viral diseases and are adept at treating the symptoms of others, the characteristics of viruses and the means by which they attack their host make cure for viral diseases elusive. Though this section, we consider the clinical implications of and the characteristics of virus, we begin by looking at the characteristics of viruses that have in common. So a virus is a minuscule, acellular infection agent having one or several pieces of nucleic acid, either DNA or an RNA. The nucleic acid is the genetic material of the virus. Being acellular, viruses have no cytoplasmic membrane. Viruses also lack the cytosol and the functional organelles that other cells do. They are not capable of metabolizing activities on their own. Instead, once viruses have invaded a cell, they take control of the cell's metabolic mechanism to produce more molecules of viral nucleic acid and viral proteins, which then assemble into a new viruses. Viruses have an extracellular and an intracellular state. So outside a cell, in the extracellular state, a virus is called a virion. Basically, a virion consists of a protein coat called a capsid surrounding a nucleic acid core. Together, nucleic acid and its capsid are also called nucleocapsid, which in many cases, it can crystallize the crystalline chemicals. Some virions, they have a phospholipid membrane, which is called an envelope, and it surrounds the nucleocapsid. The outermost layer of the virion provides the virus both the protection as well as the recognition of the sites that bind to the complementary chemicals on the surface of the host cell. So once a virus is inside the intracellular state, it's initiated, and the capsid is removed from the virus. A virus without its capsid exists solely as a nucleic acid, but is still referred to as a virus. So now that we have examined ways in which viruses are alike, we have considered the characteristics that are to distinguish different groups of the virus. Viruses differ in the type of genetic material they contain, the kinds of the cell they attack, the size, the nature of their capsid coat, their shapes, and the presence or absence of an envelope. The International Committee on Taxonomy of Viruses, ICTV, which was established in 1960s, provide a single taxonomic scheme for the viral classification and identification. The virologist has classified the viruses by their nucleic acid, the presence of envelope, the shape, and the size. So far there are established families for viral genera, but only 3 viral orders are decided. The taxonomists have not determined the kingdom, division, as well as the classes for the viruses because relationship among the viruses are not well understood. So the family names are typically derived either from the social characteristics of the virus within the family or from the name of an important member of the family. Viruses show more variety in the nature of their genomes than the cells do. Whereas the genome of every cell is double-stranded DNA, the genome of a virus may be either a DNA or an RNA. So the primary way in which scientists categorize and classify viruses is based on their type of genetic material that makes up the genome. Most of the viruses, they infect only one particular host cell. Viruses may be so specific that they infect not only a particular host, but also a particular kind of that host. So all types of organisms are susceptible to some sort of viral attacks. There are viruses that infect archaeal, bacterial, plants, protozoans, fungal, and animal cells. There is even a tiny virus which infects a large virus. Most viral research and scientific study has focused on bacterial and animal viruses instead. A virus that infects bacteria is referred to as a bacteriophage, or simply it's referred to as phage. So the scientists have determined that bacteriophages outnumber all bacteria, archaea, eukaryotes, or all of them put together. So the viruses of plants are less known than the bacterial and animal viruses. Even though the virus first was identified and isolated from a tobacco plant. Fungal viruses have been little studied. We do know that the fungal viruses are different from the animal and bacterial viruses in that fungal viruses exist only within the cell. In late 1800s, the scientists hypothesized that the cause of many diseases, including polio and smallpox, was an agent smaller than a bacterium. They called this small agent as a virus, from the Latin word which means poisonous. So the viruses are small, and that only few can be seen with a light microscope. 100 million polioviruses could fit side by side on a period at the end of a sentence. Even a small virus have a diameter of 24 nanometer, and the largest virus, which is a megavirus, is a 500 nanometer. Replication cycle of the virus, usually it results in the death and lysis of the host cell. Because the cell undergoes lysis near the end of the cycle, this type of replication is called lactic replication cycle. In general, a lactic replication cycle consists of attachment of the virion to the host cell, entry of the virion or its genome into the host cell, synthesis of the nucleic acid and the viral protein by the host cell's enzymes and the ribosomes, as well as assembly of the new virion within the host cell and the release of the new virion from the host cell. Scientists must culture the viruses in order to conduct research and develop vaccines and treatment. But because viruses, they cannot metabolize or replicate by themselves, they cannot be grown in the standard microbiological broths or the agar plates. So instead, they must be cultured inside a host, suitable host, a requirement that complicates the detection, identification, as well as the characterization of viruses. So the virologists, they have developed three types of media for culturing viruses. Media consists of mature organisms such as bacteria, plants, or animals, embryonated eggs, and the cell culture. Now that we have studied the characteristics and the replication processes of the virus, let's ask a question: are viruses alive? And to be able to wrestle with this answer, we must first recall 5 characteristics of life, which is growth, replication, self-replication, responsiveness, and the ability to metabolize, all within the structure which are called cells. So according to these criteria, viruses lack the qualities of living things, prompting some scientists to consider the viruses nothing other than complex pathogenic chemicals. And for other scientists, however, at least 3 observations that the viruses use sophisticated methods to invade cells, have the means of taking control of the host cell, and possess genomes containing instructions for replicating themselves, indicate that viruses are the ultimate parasite because they use cells to make more viruses. According to this viewpoint, viruses are the least complex living entities. In any case, viruses are at the threshold of life. Outside cells, they do not appear to be alive, but within cells, they direct the synthesis and assembly required to make copies of themselves.